All right, in this set of notes, we're going to talk cell cycle. So, in your notebook, if you'll imagine this is the center of your notebook, let me lock this in place here. You're going to have one side that's going to be information about the cell cycle, and the other side is where you are going to draw a big circle that fills up nearly the entire page. So, you can just draw a circle. I'm going to use my little bucket here to help me draw a good looking circle. All right, so let me grab a pen. All right, so there's a circle. And I'm going to put a little dot right in the middle of my circle because we're going to turn this into a pie chart. Okay, so let's start by getting some facts down about the cell cycle. So first up, let's write down the who. The who that cell cycle happens to is going to be eukaryotic body cells. And another word for body cells that we use in science is somatic cells. These are going to be things like nerve cells, bone cells, muscle cells, skin cells, basically any body cell except sperm or egg. All right, so the next step is the what. What is the cell cycle? Well, the cell cycle is a process that makes new cells. Next up, we got the when. When does the cell cycle happen? The cell cycle happens whenever an organism needs new cells. So that could be when you need to heal an injury. It could be when you need to fight off an infection, so you need to make more white blood cells. Or it could be when you need to grow. But the important thing is it only happens when cells need to divide. All right, next question would be where? Where does this happen? Well, it happens in every organism that has cells, except for prokaryotes, and it happens where those cells are. So most cells are made where they're needed, some are made in other places. All right, so last but not least, we need to talk about the steps of the cell cycle or the organization of it. And to do this, we're going to draw a flow map, and then we're going to organize that flow map onto our pie chart. So we're going to begin by titling our organization tree map Cell Cycle. Based on the tree I've drawn here, how many stages are in the cell cycle? 3. The first stage we're going to talk about, though, is actually not a part of the cell cycle. Instead, it's kind of an escape outside. This is going to be a phase called G0 phase. When you hear the number 0, what do you think about? Normally, we use the number 0 to describe when nothing is present. So this is gap 0 phase. That's what the G stands for, is gap. And in this phase, there is no cell division. That's what the nothing is for, the 0. The 0 represents 0 cell division. What is happening here is a cell doing normal cell things like respiration. If it's a plant cell or a cell that has chloroplast, photosynthesis. If it's a muscle cell, it's contracting and relaxing. If it is a root cell in a plant, it's taking up water. So that's G0 phase. For G0, we're going to add it right up here, and we're going to draw an arrow coming out of the circle, and we're going to put G0 right up here. The reason why we're drawing G0 outside of our big circle is because the circle and inside the circle represents the cell cycle. But G0 is like an escape route. If a cell does not need to divide, it will escape the cell cycle and go to G0. Turns out that cells can return to the cell cycle later on if they need to divide. So sometimes you'll hear it called a resting stage, but it's not really restful because the cell's still doing cell stuff. It's just a temporary holding place for a cell to be a normal cell when it doesn't need to divide. All right, so next stage we're going to talk about is a stage called interphase. Inter with an I means between. So this phase is between cell divisions. So interphase starts and ends where cell division stops and starts. And there are 3 stages of interphase. The 3 stages of interphase are G1, S, and G2, in that order. So let's talk a little bit more about each of these stages, and let's map them on our pie chart. We're going to set the segments of the pie to kind of be accurate to the segments that you would find on an actual, we're going to set, let me start that over. We're going to set the pie segments to be approximately equal to the amount of time that a cell would spend in those phases. So to do that, we need to find a place to start. So we're going to draw a vertical line from our center dot up to about 12 o'clock. And I'm going to write the word start. So a cell's life cycle is going to start at this line. I'm going to draw a line down to about 5 o'clock, or maybe between 4 and 5 o'clock. And I'm going to label this G1. Remember, G stands for gap. So this is going to be gap 1. And there are 3 important things that happen in gap 1. First is that the cell is going to grow. Remember, it just started life, so it's a tiny little cell, so it needs to get a little bit bigger. Second is that cell organelles are going to duplicate. Duplicate just means to copy. So things like mitochondria, chloroplasts, Golgi apparatus will all make copies of themselves in this stage. Finally, in G1, cells do normal cell activities. That's G1. Next, I'm going to draw a line kind of over towards 8, 9 o'clock. That's going to be our second segment, and that's going to be S phase. S stands for synthesis. So in this stage, the most important thing that is happening is DNA replication. That process results in 2 identical copies of DNA. Remember, this was what we modeled on our foldable. So if you need to write the word foldable somewhere in here to help remember that that's what was happening, you can do that. All right, so that takes care of S phase. So next up is G2. So we're going to draw a line from about 11 o'clock to the center. This is G2. G stands for gap, so this is gap 2. And there's 3 important things happening here. Something called a microtubule. Oops, I left out a U. So microtubules, there should be a U between the B and the L. Sorry about that. Microtubules will copy. We'll talk more in mitosis about microtubules, so don't worry about what they are yet. Next up, we prepare for mitosis. We'll talk about mitosis in just a quick second. And last but not least, normal cell activities. Cells are still doing respiration, they're still doing photosynthesis. So everything from start all the way around to here, this is all interphase. I'm going to draw me a little arrow here, and I'm just going to write interphase to remind myself. The interphase goes all the way around to here and all the way around to here. All right, so once we finish G2, now we move into our last stage. So let's go back to our tree map and let's add the last stage, which is called M phase. The M stands for mitosis. And this is cells dividing. There are 5 stages in M phase. And they start with the letters P, M, A, T, and C. I like to remember that M phase is pneumatic instead of automatic. So let's add these to our pie. I'm going to draw 4 lines to divide this last pie segment into 5, as close to equal segments as I can get. I'm going to make a note that this is M phase. So this is where cells divide. And let's label the stages in order. So after G2 comes P, whatever that means. Then M, because it's pma, A, tick, T, C. Pematic. So that's the order of the cell cycle. Once a cell divides, it goes back, it starts over as a tiny cell, it grows, and it goes through. If we don't need the cell to divide, then at some point during gap 1, the cell will escape from the cell cycle and go hang out in G0 until we need to make more copies when it'll come back in. Now you may be wondering, what does pematic stand for? Well, we'll talk more about that in our next set of notes in class. Be sure that you've completed your cell cycle and mitosis questions that go with this, and I will see you in class. If you have questions, let me know. Thanks for watching, guys.